Good morning, everyone. Today we are going to present you the results of the work Molecular Characterization of histone deacetylases and their role in type 2 diabetes. In healthy people, pancreas secretes insulin into the bloodstream to lower the level of blood glucose by allowing glucose to enter the body cells. In type 2 diabetes, either the body does not produce enough insulin or the cells resist the insulin. When glucose builds up in the blood instead of going into the cells, it can cause far-reaching health implications like heart disease, nerve and kidney damage. Type 2 diabetes is a public health problem, being one of the 10 causes of death worldwide. There is a link between gut microbiome and type 2 diabetes. Gut microorganisms ferment high-fiber foods, and this generates short-chain fatty acids such as butyrate. Butyrate is a metabolite with epigenetic effects through inhibition of histone deacetylases. Low numbers of butyrate-producing bacteria have been reported in patients with type 2 diabetes. Butyrate inhibits histone deacetylases. These enzymes regulate a variety of metabolic pathways. They remove acetyl groups from histones by modifying the chromatin structures. Their inhibition increases histone acetylation, which relaxes the chromatin structure and increases accessibility to certain genes, allowing their expression. So, butyrate raises insulin sensibility by increasing histone acetylation because of the inhibition of histone deacetylases near the promoter of the insulin 1 receptor, which increases its expression. Butyrate also increases substrate phosphorylation of insulin receptor 1 and or protein kinase B. The objective of this study is to describe the molecular mechanisms of histone deacetylases inhibition by butyrate and its effect on insulin receptor 1 expression. To achieve this objective, we make 3 research questions which we are going to describe and answer in the following slides.
Our research question number one is which human histone deacetylases inhibited by the butyrate are involved in type 2 diabetes? With this question in mind, we did a review of the different human histone deacetylases and their different functions. We found that there are 18 enzymes divided in 4 families depending on their molecular characteristics. Most of these deacetylases are related to metabolic disorders, but only 4, highlighted in the table, have a relation with diabetes or insulin resistance. To answer our question number 1, we found that of the 18 human histone deacetylases, only histone deacetylase 2, 3, 4, and 9 have been reported in academic articles in the Europe PMC related to diabetes, insulin resistance, inflammation, or metabolic syndrome. Although we already had an answer for question number 1, we decided to dig a little bit deeper to find more evidence of the role of histone deacetylases in metabolic disorders related to diabetes. Therefore, we searched for experiments that involve genes from the 4 histone deacetylases of interest. As a result, we found that each of these genes have differential expression under different pathologies, like ALS and different types of cancer, but not with diabetes. On the other hand, we found that through a genome-wide association study, a relation between type 2 diabetes and the gene related to histone deacetylase number 9 was established. So, according to the results obtained in our research, we found that only histone deacetylase 9 is involved in the epigenetic process that causes type 2 diabetes, which also answers our question number 1.
Our research question number 2: do histone deacetylases interact with butyrate? Reactome tool allow us to identify the pathways in which histone deacetylases participate and the molecules with which they interact. In the signal transduction pathway, histone deacetylases 2, 4, and 9 interact with 245 molecules. and histone deacetylase 3 with 635 molecules. In all cases, butyrate was one of the molecules with which histone deacetylase interact. Also, in the lipid metabolism pathway, histone deacetylase 3 interacts with 245 molecules, including also butyrate. Of all the tools used, reactome was the only one that allowed us to observe the interaction between histone deacetylases and butyrate, wherever this interaction could not be directly related to a disease in humans. So the answer of the question number 2 is, according to the results obtained with the reactome tool, the four histone deacetylases evaluate interact with butyrate.
The third question was, according to the information obtained so far, which mechanism of action is proposed for histone deacetylases in this context? To answer this question, we decided to learn more about the histone deacetylases structure. Through the search of these molecules in different databases, we found that the acetylases differ in their length depending on the enzyme family. In this case, histone deacetylase 2 and 3 are half the size of histone deacetylase 4 and 9. We also found that for the first pair of enzymes, the structures have conserved residues in the active site, which suggests common catalytic mechanisms. And for the second pair of enzymes, they share an equal binding site that consists of 3 cysteines and 1 histidine. Finally, only histone deacetylase 2 and histone deacetylase 4 have DNA binding sites. At the same time, in order to understand the different mechanisms of these enzymes, we look for already characterized histone deacetylases interactions. One of our first findings was that histone deacetylases work mainly by creating proteic complexes with other deacetylases and proteins. Most of these reported interactions consist in a specific protein calling for histone deacetylases for transcription regulations. For the case of diabetes and insulin resistance, specifically the insulin receptor promoter IRS1, there is still to understand how the mechanism might work, but certainly there are more proteins and histone deacetylases involved. So, to answer our third question, we found that histone deacetylases are known to form transcriptional complexes with other histone deacetylases and other proteins. These transcriptional complexes regulate gene transcription without direct binding to DNA. Conclusions. Histone deacetylase 9 could be involved in the negative regulation of genes responsible in glucose metabolism, and with the presence of butyrate, these histone deacetylases could be inhibited and allow histone acetylation in the vicinity of IRS1 promoter, which later might increase its expression. Evidence seems to indicate that variants of histone deacetylase 9 are present in patients with diabetes or insulin resistance. However, this protein, despite being located in the nucleus, has no DNA binding sites. But because histone deacetylases can be a part of different protein complexes, we suspect that this is the way this enzyme accomplishes this binding. Finally, the use of histone deacetylase inhibitors such as butyrate could be used for the treatment of diseases such as diabetes.